Hi, everyone. It's me, Professor Willis, and I'm here to talk to you about your third lab, which is on osmosis and diffusion. I'm going to give you a little basics first about osmosis and diffusion, and then we'll jump into the details of the lab. So today's lab, or this week's lab, is on diffusion and osmosis. And diffusion is the movement of molecules from a high concentration to low concentrations. What I always tell my students is things in nature, they don't like to be crowded. They like to spread out. So if you can see here in this animation, the red dots are molecules, and you'll notice they diffuse through the screen. So they start off crowded in a high concentration, and they move to low concentrations. It occurs all the time in cells. So cells are constantly moving things in and out of their cells to sometimes eliminate waste, to take in nutrients that they may need, and they all follow the same basic principle. Things will move from high concentration to low concentration. So if inside the cell there's a low concentration of items and outside the cell there's a high concentration of those items, then those items are going to naturally want to move from high to low. So they'll move into the cell. Now, of course, the cell sometimes has to use special tools in order to do that, but it still follows the same basic principle, high to low concentrations. So who is doing all the regulation for this movement? Well, if you can remember from this cartoon image here, this is a picture of the cell membrane. So the cell membrane of the cell is the one that's performing the movement and allowing things to come in and out of the cell. All cells have a cell membrane, whether it be a plant cell or an animal cell or a bacterium. Any of those will have cell membranes, so they all allow for the regulation of materials in and out of the cell. So just a little review of the cell membrane. It's made of two semi-permeable phospholipid layers. So semi-permeable means that some things are allowed through the membrane, but not all things, okay, semi. And phospholipid, well, if you remember what a lipid is, it's a fat, and it has a phosphate head, which is a PO4 group, a chemical group. So it's made of those two layers, and you can see from the picture, the phosphate heads are on the outside, and then the lipid tails are in the inside. What's crazy about this layer is if usually like you're a larger molecule, you can't get through the layer. You have to use special proteins or channels to let you through. But if you're small, you can squeeze through the membrane. Water will do this sometimes. However, water does sometimes use these things called aquaporins that are channels that allow water to come in and out of the cell. But it's the same basic principle. There's two layers of lipids, phospholipids, and they're the things that are regulating what is coming in and out of the cell. So a little review on what is osmosis. So osmosis is simply the diffusion of water. So when you have water moving across a semi-permeable membrane, we call that osmosis. And so today's lab, this week's lab, is on looking at how water moves across membranes when you put them in different concentrations of solutions. Osmosis, water, works the same way. It always likes to go from high to low concentration. So keep that in mind. Why does the water like to move to different places? It will follow the magic rule, high to low concentration. So this is a lab that biology students often do in experimenting with osmosis. There's this material that you see here in the picture. It's called dialysis tubing because it is used in dialysis, and it's a semi-permeable membrane. And so what a lot of teachers will do is they have this tubing, and you can change the concentrations of the materials in and out of the tubing. And so when you do that, then you can observe how much water or other solutions if you're testing are coming in and out of the bag. And I wanted to show you this cartoon because this is kind of the classic experiment. So you can see the yellow is the dialysis tubing and it's placed in a beaker. What you can do is change the concentrations of the solutions in and out of the bag. So for example, in this situation, the bag is 0.2 molar and outside the bag in the beaker is 0.8 molar. And so once you sit the bag in the beaker, you can then observe what is going on with the water. Is it going to move into the bag or out of the bag based upon the different concentrations of the materials in both solutions? Okay, so here is some good information because you're going to use this in your online lab. So in that previous experiment, if the bag gets heavier, so say you weigh it before and after the experiment, right? And you notice that after the experiment, the bag got heavier. That means water entered into the bag. And that means that you placed your bag in what's called a hypotonic solution. Now, the way I remember this is hypo kind of looks like hippo, and hippos tend to be larger, right? So if your bag gets larger, heavier, that means water entered the bag and you place that bag in a hypotonic solution. Now, say you take the bag out and you notice that the bag got lighter. So you weighed it after it sat in that material and the bag got lighter. That means that water left the bag and you placed your bag in what's called a hypertonic solution, right? All right, so say you take your bag out and you weigh it, and after it's the same weight as what it was before. That means there was no real, there was movement, but the movement is equal, so it was in equilibrium, and we call that an isotonic solution. So get comfortable with those words in your virtual online lab. You're going to be looking at what happens to cells when you put them in these different solutions. So the challenge for this week's lab is under what conditions do cells gain or lose water? So you're going to be subjecting different cells into solutions and you're going to see whether or not they will gain or lose water or maybe nothing at all. In this week's lab, you're going to go through 3 different components to learn about the basics of osmosis and diffusion. The first part of the lab is simply going to the website that's listed on the paper, and you're going to learn about diffusion. So you're going to see some animations that show how cells move materials in and out of their system. And then all you have to do is answer the questions in part 1, of course, using complete sentences, and then you're done with part 1, diffusion. Part 2 is similar to part 1, but in part 2, you're going to be investigating specifically osmosis, or the movement of water in and out of a cell. Remember, osmosis does not require the use of energy. It's a passive transport mechanism. And so cells, obviously, they require water, so they need to be able to move it in and out of their system. So just like you did in part 1, you're going to go to the website listed there on the paper and work through that website and answer the questions that are on your paper. The last part of the lab for this week is an actual online investigation. So you're going to be going to this third website here, and you are going to see how changing different sugar solutions, how that affects the mass of these beakers and solutions. So each of these beakers are going to have different sugar solutions. You're going to see what happens to the weights. You're going to be calculating weight changes and also percent changes with those weights. And then lastly, you're going to construct a graph. Now the graph, you can do it one of 2 ways. Number 1, you can graph your data on a piece of paper, take a picture of it, and submit it onto the paper. Or if you want to do the graph via Excel, that's fine. Just go ahead and use Excel and then take a screenshot of that and then put it into the lab report.
Lastly, you make a scientific claim about what you discovered from this lab and what were some data trends. Like I stated last week, remember there's no formal lab report needed. You just need to work through this lab, answer the questions that are on the paper, and then submit it to Blackboard by next week's due date. All right, everyone. Thanks, Biology 103, for listening. Good luck, and I will see you next week.